Hello, my name is William Stifler, and this is my project on basically seeing what epibionts live on the hybrid snake population right behind the Zeta complex on Eckerd College. Over the summer, I started reading about snake fungal disease, the causative agent of which is Ophidiomyces ophiodiicola. And what that is a fungus that gets into the skin of snakes, and it kind of eats the keratin. And if it gets through the skin and the snake can't shed it off, it starts eating the snake's organs, and then the snake dies. And that's really upsetting, and we don't want that to happen. And over the summer, I read several works that kind of showed that the cutaneous microbiome may have an inhibitory effect towards this fungus, because when they grow it on agar, fungus that is, and they swipe certain bacteria through it, for example, Morganella morganii, the growth of the fungus is inhibited. That is kind of the end goal. That's the NSSRP that I applied for this summer. But to be able to do that, first you should start with seeing what bacteria you have. So what I did is I went out and I swabbed the snakes in the pond, and I used about 4 different methods to see what I had. The first is methylene blue stain, which kind of tells you what the bacteria you're working with looks like. Then I did the gram stain, which tells you whether or not you're working with bacteria that has a thick peptidoglycan membrane, or one that has peptidoglycan on the underside, and then something else on the outside of the cell. Then there's a catalase test, which tells you whether or not the enzyme catalase is present inside of the bacterium. And finally, a motility test, which tells you whether or not the bacteria is motile, at least in water, the way that I performed it. Between all of these tests, I was able to kind of tell what families I was working with, not necessarily to the genre. And I did this, of course, with the caveat of saying I did it in tandem with other snake family or other families of bacteria that have been found on other species of snakes, as it's been shown that that kind of microbiome doesn't really vary even between different snakes in different regions. That hasn't been done on an aquatic snake before, so I could be wrong. However, it does seem that these families right here, Aeromonadaceae, Enterobacteriaceae, Nocardioidaceae, what is that? Oh, Oxalobacteriaceae and Xanthomonadaceae. I hope I pronounced at least one of those right. Those are probably some of the ones I'm working with. Nothing to the genus level yet, or at least not with the confidence that I would mention it here. But we do have, or I have about 15 specimens cataloged so far, and about 25 on the back burner. So I'm excited to get through all those. Obviously, if this NSSRP grant goes through, what I would like to do is start doing genetic testing of the bacteria to see what I have. And then, once I have bacteria isolated, if I can also do genetic testing and make sure that I'm able to culture some of the snake fungal disease agent, which I'm just going to abbreviate OO for now, then I can do those exact same fungal challenge assays, which is kind of what I want to do in the first place, because unfortunately the snakes in Zeta Pond experience a really high rate of SFD infection. I've maybe pulled out 2 this year out of 40 that didn't have some symptoms of snake fungal disease. And yeah, because of their basking habits, they're extra at risk because they don't spend much of the day basking compared to other snakes, especially not when you live on a campus with some of the highest bird diversity, and birds like to eat these snakes, of course. That is the introduction to my presentation, and I look forward to any questions you have. Thank you very much for coming.